Good afternoon. My name is Russ Smith, and I'm going to be presenting on the role of genomics in solving the sustainable energy crisis. First of all, the need for sustainable energy. There's a great need for the solution to the global energy crisis that we're facing today. We have a growing demand for safe and sustainable alternate fuel sources, especially in the Department of Transportation. First-generation biofuels such as ethanol and biodiesel have been researched extensively, but the problem is that they're often not sustainably sourced, and they're processed in ways that are more harmful to the environment than fossil fuels. This has led researchers to dive deep into second-generation biofuels, which have proven to be an emerging solution to this problem. Today, we're going to be focusing primarily on lignocellulosic biofuels as a promising prospect for the energy crisis. So why lignocellulose? Well, lignocellulose is a lignin-cellulose complex found in the cell walls of woody and fibrous plants. According to Himmel and his research team, cellulosic biomass is an abundant and renewable resource, and research has shown that lignocellulosic biomass represents the most scalable and environmentally stable biofuel on the planet, according to Hill et al. In their research conducted and published in 2006. So lignocellulose is comprised of three major components: cellulose, hemicellulose, and lignin. So cellulose is the most abundant component of lignocellulose and is composed of beta-1,4 linked chains of glucose molecules. It also contains hydrogen bonding, which causes resistance to degradation. Hemicellulose is the second most abundant component and is composed of 5 and 6 carbon sugars, such as arabinose, galactose, glucose, mannose, and xylose. Finally, lignin is composed of 3 alcohols. For our sake, we'll call H, G, and S. Cellulose, hemicellulose, and lignin form structures called microfibrils, which are combined to form macrofibrils, which mediate structural stability in the plant cell wall. So, how are lignocellulosic biomasses utilized to create biofuels? Well, first, solar energy is captured through photosynthesis and stored in the cell walls stored in the cell wall polymers in the form of cellulose, hemicellulose, and lignin. The biomass is then collected and it's deconstructed through chemical or enzymatic treatment into component sugars. These component sugars are converted into biofuels using fuel-producing microorganisms. Now, where does genomics come into this? Well, in terms of genomics, we're going to be focusing on fuel stocks with modifiable traits, where enzymes can be engineered for biorefining. So specifically, we'll look at the plant switchgrass. Due to its abundant productivity, adaptability to high growth in poor soil, low nutrient and water requirements, and its capacity to recycle nutrients very efficiently. There has been extensive study done on switchgrass using whole genome sequencing. We'll look at one study in particular, conducted and published by Sharma and Sharma in 2012, called a genome-wide survey of the switchgrass genome structure and organization. By way of overview, switchgrass is a perennial grass, a promising biocrop, and the target of whole genome sequencing. For this study, 2 bacterial artificial chromosomes, we'll call BAC libraries, from the AP13 clone of switchgrass were constructed to gain insight into the structure, organization, comparative genomic studies, and to assist with genome assembly. Each library comprised of 101,376 clones, with average inserts of 144 and 110 kilobases. There were a total of 330,297 high-quality BACN sequences. We'll call these BESs. Generated in the study, which accounted for about 16.4% of the switchgrass genome. Analysis of the BES identified about 297,099 known repetitive elements over 50,000 simple sequence repeats, and 2,528 novel repeats elements called switchgrass repetitive elements. Comparative mapping demonstrated high levels of synteny, or collinearity, with grass genomes of sorghum, rice, and maize. Further, within this study, there was an annotation done and gene ontology analysis. This gene ontology analysis was performed to identify the vast biological functions encoded by the switchgrass genome. The gene ontology analysis was performed with regard to three major ontological categories. The three we discussed in class: molecular function, biological process, and cellular location. Out of the over 300,000 BES, 5,052 could be associated with at least one geoterm. Specifically, 4,507 reads were assigned to at least one of the 377 molecular function categories. 3,244 reads were annotated to at least one of the 259 biological function categories. And 1,144 reads were associated with at least one of 80 cellular locations. This data is represented in these graphs here. First, in section A, you see the cellular functions category, and we see that the vast majority either fall into the category of membranes, which accounted for 37%, or protein complexes, which accounted for 21%. Section B, biological processes, 31% fell under the category of other metabolic process, and 25% fell under the category of protein metabolism. Finally, there were a number of categories that were equally distributed among the molecular function category, including nucleotide binding, metal ion binding, nucleic acid binding, hydrolase activity, and catalytic activity. Another section of this study focused on the analysis of microcollinearity using full-length BAC sequences. In this section, 47 randomly selected BACs were sequenced to essentially full length using Sanger's method. They discovered corresponding orthologs for about 370 gene loci could be identified from rice, 363 could be identified from maize, 357 could be identified from sorghum, and 336 could be identified from brachipodium. And this is the pictorial of these findings, of this data. We don't have time to get into it right now. But finally, we're going to talk about some of the challenges that were faced in this study and some of the areas that are available for future research and study. They mentioned that the largest difficulty faced in this study was discriminating between paralogs, orthologs, and homologs in these regions when they were sequencing the switchgrass genome. And further research of the switchgrass genome can be identified and can be found at switchgrassgenomics.org. On this website, they specialize in researching increasing conversion efficiency by reduced lignite cation to increase microbial access to cell wall carbohydrates, genetic variation of lignin for optimal conversion, identifying cell wall and stress-related genes in switchgrass, and using whole genome expression profiling the transgenetic lines for engineering stress tolerance. In conclusion, we have seen that switchgrass and many other lignocellulosic biomasses can offer a solution to the sustainable energy crisis faced globally today. Thank you very much.